Hello and welcome to Plant Science Day virtual barn exhibit. I am Doug Brackney and I'll be presenting work today on how mosquito feeding behavior impacts disease transmission. So there are over 500 known arthropod-borne viruses or arboviruses, of which approximately 100 of them cause disease in humans. And the ones that cause disease in humans are primarily from three families: the alphaviridae, the flaviviridae, and the bunyaviridae. We'll be talking today mostly about flaviviridae, and these include viruses such as dengue virus, yellow fever virus, Zika virus, and West Nile virus. Arboviruses are transmitted globally. They're on every continent except for Antarctica for obvious reasons. One thing that you'll see from this is that there's a lot of viruses that are transmitted in temperate zones. However, the majority of these are tick-associated arboviruses. With a couple notable exceptions, such as West Nile virus and Japanese encephalitis virus, which can be found in more temperate regions. A large majority of these viruses are actually found in the tropics. And this really has to do with the fact that the primary vector for a number of these arboviruses is Aedes aegypti. As you can see on the map in the bottom corner, this is a environmental suitability map for potential Aedes aegypti breeding. You can see that it's pretty much confined to the tropics. It likes nice warm and moist climates. And the reason I bring up Aedes aegypti is because it is the best vector to transmit arthropod-borne viruses, and it's the main culprit for the transmission of dengue, yellow fever, Zika, and chikungunya viruses. And the reason it's such a good factor is because it's anthropophilic, so that means that it really likes to feed on humans. In fact, 95% of its blood meals are acquired from humans. Also, it's endophilic, meaning that it likes to live, breed, and feed in close proximity to homes. So it really it lays its eggs in artificial breeding containers such as discarded tires, water vessels for holding water, and so it's really in close proximity to humans. Another thing that makes it a very good vector is that it feeds frequently. And I really want to stress this because that's going to be a topic of what we're talking about today, is the frequency at which these mosquitoes feed. And they'll feed every 2 to 3 days in nature. And this really facilitates effective transmission of a lot of these arboviruses. And then another thing that makes these Aedes aegypti so prolific and resilient is the fact that they do this thing called skip oviposition, where they actually lay their eggs in multiple different containers as opposed to putting them all in, say, a single basket. So they might lay a couple eggs in a tire here, a couple eggs in another container over here, and this really buttresses them against human behavior effects, such as dumping out a discarded tire or drinking water out of a water container and reducing all the water in there, so the eggs then will die. By spreading it to multiple different containers, it really prevents it from the effects of human behavior. So I want to introduce you to this one concept right now called the Extrinsic incubation period, or the EIP. And this is the time from infection to the time of transmission. So during the normal course of infection, a virus will take a blood meal off of an infectious host. It could be a bird, could be a human, could be a horse. The blood from the host, along with virus in it, gets transported up into the gut of the mosquito. We call it the midgut. And what you have occurring is the virus then enters the midgut epithelial cells and begins replicating inside those cells. And then as time passes, eventually the virus has to make its way out of the gut, so it has to go out of the other end of the gut cells and make its way into the open circulatory system. So once it disseminates out of the gut into the open circulatory system, the virus then will eventually make its way to the salivary glands, establish infection there, replicate again, and then ultimately then be released in a subsequent blood meal. So what do we know about blood meals? So I have a figure here that basically shows sort of the genes that are altered during blood meal acquisition. So if you look right here in this column, you have mosquitoes that have been provided a sugar meal, and over here those that have been provided a blood meal. And then each one of these bars right here going across represents a different gene that was assayed in this experiment. And red or green then tells you whether a gene is either upregulated, so there's more production of it, or downregulated, so there's less production of it. And what you can see is for all these different genes that are listed along this vertically, you can see that there's a huge physiological change in at least the gene expression when it comes to mosquitoes in sort of the sugar-fed state. So when they're actually just emerged and they're maybe feeding on flowers and stuff, versus those that then have been taken a blood meal. You have a huge shift in the array of genes that are being expressed. So we know that blood meal results in sweeping physiological changes in mosquitoes. And this is quite evident when you just look at a gut and what's happening. So when you have a sugar-fed mosquito, you see you've got a very translucent, sort of thin membrane or tissue inside the gut. But when you take a blood meal, this tissue expands to about 10 times its size. The mosquito becomes much heavier, and it has to quickly digest this blood meal so that it can then escape predators and fly. This really is a huge event in the life of the mosquito. And what you can see is that you get this huge blood meal here, the midgut expands, and eventually over time, you see that the blood meal is digested, and the gut gets back to looking the way it did at the beginning. So one thing I want to talk about this is, so as I said earlier, the midgut has these midgut epithelial cells, which are represented here by these sort of peachish color little cells with little nuclei on the inside. And this is the midgut epithelial cells. It's a single cell layer thick, and then it's supported by this underlying tissue called what we call the basal lamina. And that provides support and protection to the midgut epithelial cells. And so what we know about the basal lamina is that it's a dense network of these different proteins here, nitogen, percolin, laminin, collagen, and then it has a pore size of about 10 nanometers. And so this pore size provides enough room for micronutrients to go across the membrane, across the basal lamina, as well as oxygen and stuff like that. But at the same time, it prevents larger molecules from passing from outside the gut into the gut. And this is important though, because what we know is that the average arbovirus is about 50 nanometers in size. So this has been a big question in our field, is how does a virus get from these midgut infected cells out of the gut and all the way to the salivary glands that we talked about earlier? So we think it actually has to do with the mosquitoes taking a blood meal. So we took some mosquitoes that were uninfected, gave some a blood meal, others did not, and then we did scanning electron microscopy. And this is a very high-powered form of microscopy. We can look in detail at the substructures of the gut. So we've done this for unfed mosquitoes 24 hours post blood meal and 72 hours post blood meal, and we looked at 300x and 4000x. I would really like to draw your attention to the 4000x column here. As you can see, in the unfed mosquitoes, you really have this basal lamina is, all this stuff here, and it just sort of has a ruffled, relaxed look to them. And there's no breaks, it's a continuous layer. However, after a blood meal, we see that we start getting these big cracks forming in the surface of the basal lamina. These represent muscular tissue here on the outside, and this is the basal lamina here, and inside there is then this big crack. And then by 72 hours, once the blood meal has been beginning to be digested, and you're starting to then get repair of the basal lamina, and all of a sudden this crack starts becoming a lot smaller and more closed, so particles can go through. So the thought is that what's happening is the virus is getting into the gut, replicating, and then it can eventually pass through the basal lamina because of these cracks that are introduced. However, in the laboratory, we often assay this thing called vector competence. And so this is the ability of a vector arthropod to become infected with a pathogen, permit replication, and ultimately transmit the pathogen to a naive host. And the way that this is normally modeled in the laboratory is we take a bunch of mosquitoes, we take virus that we grow in cell culture, add it to some blood, offer it to the mosquitoes, and then at subsequent times post-infection, we look at mosquito infection rates. So we'll harvest tissue, mid-gut, other tissue, salivary glands, or saliva to look at infection and disseminated infections and whether the mosquito is transmitting. However, that really doesn't mimic what's happening in nature, right? We like I said earlier, Aedes aegypti is known to feed every 2 to 3 days. However, those experiments, the typical vector competence experiments, have been going on for 100 years and never taken into consideration what happens when a second blood meal, which causes these large physiological changes, what effect that's going to actually have on the virus. So we decided to test that and look at this in a very simple experiment. We provided an Aedes aegypti with Zika virus blood meal, separated them into 2 groups, waited 4 days, gave half of them a non-infectious blood meal, so no virus this time, just regular blood, and then held them for an additional 3 days. So 7 days post-infection, we then assayed for Zika virus infection in the gut and also in for disseminated infection. And so when we do this, as you'd expect, your mid-gut infection rates would be the same between the 2 groups because they were fed the same initial infectious blood meal. However, then when we looked at disseminated infections, so virus outside of the gut, we found that there was a much higher percentage of mosquitoes that had virus outside of the gut than those that did not in the single fed group. So this is showing that this process of taking an infectious or non-infectious blood meal at later time points can really facilitate the virus's ability to get out of the gut. And so we did a time course and we just looked at this is days post-infection, so initial blood meal, and then we held them for 4 days, and then we assayed them at 5, 6, 7, 8, 9, 10, or 10, 12, and 14 days post-infection to see what was happening here. And what we noticed is that really early on, after that second blood meal, we see this huge increase in the amount of viruses that is disseminated in the mosquitoes that were provided at double feed versus the single feed. Ultimately, the single feed does catch up at later time points, but it's these early time points that we're really interested in. And then when you actually look at Zika virus transmission, so virus in the saliva, we actually see a significant increase in the percentage of those that are actually transmitting Zika virus at these early time points. We then went on to see, okay, if this is unique to Zika virus and Aedes aegypti, or if this was consistent among other viruses. And so we looked at dengue virus, another flavivirus transmitted by Aedes aegypti, and again we see an increased rates of dissemination. We tested with chikungunya virus to see if it was chikungunya virus is an alpha virus. And so we wanted to see, okay, is this phenomenon that we're observing unique to flaviviruses, or is this something we observe in other arboviruses as well? And again, we see increased dissemination rates for chikungunya virus and increased transmission rates for chikungunya virus. So what we're observing here is that this second blood meal definitely seems to be facilitating virus escape from the gut at these early time points. So the working model is that under a single feed scenario, virus comes in during a blood meal, you get some cracks that are formed right here and here. Virus starts replicating, spreading cell to cell along the mid-gut epithelial cells, and the cracks start sort of recovering. The virus continues to spread cell to cell, and then eventually at a later time point it finds one of these cracks that wasn't completely fixed, and the virus is able to exit out of the gut. However, in a double feed scenario, you get the initial infection as before. You get cell-to-cell spread, but then an additional blood meal is added, and when you do that, you get the introduction of new cracks, and the virus is then able to exit the gut at an earlier time point compared to the single feed group. So why do we care about this? So to sort of model this and what the larger effect this would have during an epidemic, we use this concept called vectorial capacity. And this is the overall ability of a vector species at a given location at a specific time to transmit a pathogen. And so quantitatively, vectorial capacity can be defined as the number of new mosquito infections arising from one infected person. And there's a number of different factors that go into this, such as vector biting rate, the competency of the host, daily survival of the mosquito, vector lifetime. These all get input into this vectorial capacity equation. So when we start looking at this and putting our data in, one thing that we notice first off is that we have a reduced extrinsic incubation period. Now again, that is the time from infection to time of transmission. And we see that in the double feed group, it's about 3 days earlier than what we see for the single feed group. So we know that these viruses are actually being transmitted earlier in the double feed group compared to the single feed group. So what that means is that under field conditions, if you have a single fed mosquito, it'll become infected and then it'll take 10 days before then it feeds. And this is how we've commonly modeled this in the laboratory, right? However, what we're saying is that now a mosquito takes a blood meal and then 3 days later it takes another non-infectious blood meal. So sort of what we're modeling in the laboratory and what is representative of what actually is happening for Aedes aegypti in the field. Then 4 days later, so for a total of 7 days, then they're able to transmit earlier. If you then, and this is important too, because if you factor in then the survival of the mosquitoes in nature, there's a lot of things, predators, people swatting them, differences in temperature and desiccation. You have, a population that is eventually decreasing over time. So if they're able to transmit earlier, then you actually have you have an increased number of infectious mosquitoes that are available to bite and feed on individuals. And so the thought is that this increased transmission capabilities at these early time points then can really explain the explosiveness of recent arbovirus epidemics such as Zika virus and chikungunya virus here in the New World. And this can really be crystallized with this by equating the vectorial capacity, and basically find that in the double feed group you increase vectorial capacity by 1. So you would actually have, as opposed to if you had an infected individual, under a single feed scenario you could potentially model and say that 3 potential mosquitoes would become infected from that individual. Under the double feed scenario, this would actually then be over 4 individual mosquitoes would become infected. So just increasing the number of mosquitoes that are infectious to then transmit. And if you take this over multiple generations, this begins to expand quite exponentially. So with that, I would like to thank a number of people here at the Ag Station, including Angela Bransfield, Dr. Phil Armstrong, who is a co-author on this study with me, Dr. Andrea Gloria Soria, Mike Masinchek, John Shepard, Mike Thomas, Maria Correa, and PJ Conway, all at the Ag Station, and contributed and assisted in this work and couldn't have been done without them. The modeling aspect was done by graduate student Hannah Ehrlich at Yale School of Public Health, and at her time, her advisor was Dr. Virginia Pitzer. And then this was really supported by the Northeast Regional Center of Excellence for Vector-Borne Diseases grant that we have that was funded by the Center for Disease Control and Prevention. And with that, I'd like to thank you and have a good day.